Assalamu alaikum. In this video we are going to talk about the concept of operon. Lac stands for lactose, which is a disaccharide sugar, and operon is a system. So operon is a system governed by the presence of lactose. A more formal definition of an operon is a unit of genetic material that functions in a coordinated manner by means of an operator, a promoter, and the structural genes. Operons are mostly found in prokaryotes and are absent in eukaryotes. They are polycistronic, which means that a single mRNA transcript can code for multiple proteins. Bacteria uses the concept of operons to minimize energy wastage, as resources are limited in the environment, so the bacteria does not want to waste enzymes and proteins coding for lactose metabolism when lactose is not present in the environment. This is the structure of the operon. We have a promoter, an operator, a repressor, and the structural genes LAC-Z, LAC-Y, and LAC-A. An easy way to remember this is PROG, which stands for promoter, repressor, operator, and then genes. If you take a closer look at the structural genes, LAC-Z encodes for beta-galactosidase. Beta-galactosidase is an enzyme that cleaves the lactose molecule into glucose and galactose so that it can be easily metabolized by the cell. LAC-Y codes for permease. Permease is a membrane channel protein that binds to the cell membrane and increases the permeability of lactose into the cell. LAC-A codes for trans-acetylase. The role of trans-acetylase is not yet completely discovered, so we can say that the major enzymes or proteins playing role in lactose metabolism are beta-galactosidase and permease. If we move upstream from the operon, we have the CAP site. CAP stands for catabolite activator protein, and this site plays a role in lactose metabolism in the presence of glucose. If we move further upstream, we have LAC-I and the promoter for LAC-I. LAC-I is a regulatory gene, and it encodes for LAC repressor or LAC inhibitor, which then binds to the operator and the whole operon becomes switched off. When the LAC repressor is not bind to the operator, the operon is switched on. Now we are going to discuss operon in more detail and discuss the mechanism of how lactose turns the operon on and off. Now usually lactose is not present inside in the environment, so it is not present inside the cell. So the operon is in an off state. Now what does that mean? It means that the repressor is bind to the operator, and it blocks the RNA polymerase. The RNA polymerase binds to the promoter, but it cannot move ahead to transcribe the genes, the structural genes, which are LAC-Z, LAC-Y, and LAC-A. So in this condition, in the absence of lactose, the operon is off. Now, what happens in the presence of lactose? When lactose is present in the environment, the cell takes up the lactose, and non-lactose is present inside the cell. Now, along with lactose has an isomer, which is allolactose. Now, in the presence of allolactose tends to bind to the repressor. Repressor has a small site for the binding of allolactose, so the allolactose binds to the repressor in this manner. Now, what happens by the binding of the allolactose? The binding of the allolactose causes a distortion in the shape of the repressor so that it cannot remain attached to the operator. The repressor cannot remain attached to the operator, and it is now removed by the binding of allolactose. Now the operator is free. So in this case, the RNA polymerase can now bind to the promoter region and then move ahead transcribing the genes, structural genes LAC-Z, LAC-Y, and LAC-A, reducing the mRNA transcript, which can then be translated to produce beta-galactosidase, permease, and trans-acetylase so that lactose metabolism can take place. And this is how the binding of allolactose to the repressor switches on the operon. Now we are going to discuss the effect of glucose on the level of transcription of operon and how glucose affects the rate of RNA polymerase. Now the first scenario we are going to look at is when glucose is absent and lactose is present. So then for the operon to be operational, these two conditions must be met. So in the presence of lactose, the usual process takes place. Allolactose binds to the repressor and the repressor detaches from the operator, and now the operator is free. So the RNA polymerase can now move freely and transcribe the structural genes. But in the absence of glucose, an additional regulatory process takes place. What happens is that in the absence of glucose, cell is in a starvation condition. So ATP is being consumed and cyclic AMP is being produced. Cyclic AMP binds to the CAP protein. CAP stands for catabolite activator protein. Now we have discussed that upstream of the promoter of the operon we have the CAP site. Now in this condition, this site comes in play. So cyclic AMP binds to the CAP protein and forms a complex. Now this complex binds to the CAP site and it increases the level of transcription of RNA polymerase. So the activity of RNA polymerase increase and the level of transcription increase as a result of this. So more genes are being transcribed at a faster rate. It means that more beta-galactosidase will be produced, more permease will be produced, and more trans-acetylase will be produced at a given time period. So the lactose metabolism will increase as the concentration of enzymes and proteins coding for lactose metabolism will increase in the cell. So what happens in the presence of glucose? Now we know that glucose is a more preferred energy source of the cell, and glucose is always preferred over lactose. So when glucose is present, cell is going to prefer glucose as an energy source, and it is going to metabolize glucose rather than lactose, because metabolism of lactose requires more energy, as lactose is a disaccharide sugar. It needs to be broken down and many more processes are to be done for its metabolism rather than glucose. So the cell is always going to prefer glucose as an energy source. In the presence of glucose, what happens is that ATP is being produced. So cyclic AMP is not present in the cell. So what happens by the absence of cyclic AMP is that this complex cannot bind to the CAP site. Now when this complex does not bind to the CAP site, it decreases the level of transcription of RNA polymerase, and it slows down the level of transcription to much extent, so that these proteins and enzymes are not being produced, as the mRNA transcript is being produced at a very slow rate because of the lack of binding of the CAP plus cyclic AMP complex to the CAP site. Hence, in this case, more glucose metabolism will take place and lactose metabolism will be slowed down due to the lower transcription and then translation of beta-galactosidase, permease, and trans-acetylase. So in this way, the bacterium and other prokaryotes regulate their gene expression. I hope this video has helped you build up the concept of how operon works and what is the role of catabolite activated protein and how it increases or decreases the level of transcription. And we have also discussed the effect of glucose on the level of transcription of operon. If you have any confusions regarding any of the concepts we discussed, you can rewind and watch previous parts of the video. And thank you for watching this video.